Decoding Life, an introduction to genomics. Genomics is the study of genomes. Genomes are the complete set of DNA within an organism. DNA is the blueprint of life. It contains instructions for building and maintaining an organism. Genomics helps us understand how life works. It also helps us understand and treat diseases. Genomics has revolutionized biology and medicine. It allows us to read the book of life itself. By studying genomes, we can learn about our ancestry, our predisposition to diseases, and even the evolutionary history of life on Earth. The field is rapidly evolving, with new discoveries emerging all the time. Genomics has the potential to transform healthcare. By understanding our genes, we can develop personalized medicine. This means tailoring treatments based on an individual's genetic makeup. Genomics also plays a crucial role in drug discovery and development. Furthermore, genomics is essential for understanding biodiversity. By comparing the genomes of different species, we can trace their evolutionary relationships. This knowledge helps us conserve endangered species and manage ecosystems effectively. Sequencing the blueprint NGS and WGS. Genome sequencing is like deciphering the order of letters in the book of life. Next-generation sequencing, or NGS, has revolutionized this process. NGS allows us to sequence DNA faster, cheaper, and more accurately than ever before. Whole genome sequencing, or WGS, is a type of NGS that decodes the entire genome of an organism. NGS involves fragmenting DNA into small pieces. These pieces are then sequenced simultaneously. Bioinformatics tools are used to assemble the sequenced fragments. They are put together like a jigsaw puzzle to reconstruct the entire genome. WGS provides a comprehensive view of an organism's genetic makeup. It allows us to identify mutations, variations, and other genetic changes. This information is crucial for understanding disease, developing new treatments, and studying evolution. The advancements in NGS and WGS have led to a significant decrease in sequencing costs. This has made genomics more accessible for research and clinical applications. As sequencing technologies continue to improve, we can expect even greater insights into the complexity of life. Finding needles in haystacks. Gene prediction. Genes are the functional units of DNA. They contain the instructions for building proteins, which carry out various functions in cells. Gene prediction is the process of identifying genes within a sequenced genome. It's like finding needles in a haystack of DNA sequence. Gene prediction algorithms use computational methods to identify potential genes. These algorithms look for patterns in the DNA sequence that indicate the presence of a gene. These patterns include start and stop codons, promoter regions, and other regulatory elements. Several tools have been developed for gene prediction, each with its strengths and limitations. These tools use different algorithms and statistical models to identify genes. Some popular gene prediction tools include Augustus, GeneMark, and Snap. Gene prediction is a challenging task. Not all genes are easily identifiable by algorithms. Experimental validation is often required to confirm the presence and function of predicted genes. However, gene prediction algorithms remain essential tools for genome annotation. Making sense of the code genome assembly and annotation. Genome assembly is like putting together a jigsaw puzzle without a picture on the box. Once a genome is sequenced, the pieces need to be assembled in the correct order. Genome annotation is like labeling the pieces of the puzzle. It involves identifying and describing the different parts of the genome, such as genes, regulatory regions, and repetitive elements. Genome assembly relies on sophisticated algorithms that align and merge overlapping DNA fragments. The goal is to reconstruct the original sequence of the genome as accurately as possible. Annotation involves identifying the locations and functions of genes and other genomic elements. This is done by comparing the sequenced genome to databases of known genes and regulatory sequences. Genome annotation provides a roadmap for understanding the organization and function of a genome. It helps us identify potential drug targets, understand disease mechanisms, and study evolutionary relationships. Several tools are available for genome assembly and annotation, such as Proca, Rast, and Maker. These tools automate the process and provide comprehensive annotations. As our understanding of genomes grows, annotation tools are constantly being updated and improved.